everybody, how are we doing? So now we'll be discussing on our second case study, which is the question which was forwarded is this one. Okay, what will happen if the cow, which is in average 300 kilogram, ate carrots of 2% of its body weight? So 2% of its body weight means it's 6 kg. Perfect. So let us share our finding for today. And shall I start first?
Yes.
Okay then. So I managed to find the chemical composition of the carrot, including of the functional properties and the processing of carrots a bit. So because for this case study we'll be focusing much on the carbohydrate. So I managed to find only the carbohydrate composition in the carrot. So it is said that it has 10.6% of carbohydrate. And another form of carbohydrate is in the form of fiber. So the crude fiber is 1.2%, in which it contains cellulose, which is 89.9%, hemicellulose 9.14%, lignin at 2.48%, and pectin. So we know the differences between cellulose, hemicellulose, and lignin, whereby hemicellulose is the one which is being digested the most easiest, and then cellulose comes second, and then lignin is the most tough carbohydrate. And then not to forget, it has fats. It has 0.2% fats. So we know the cell wall has the pectin, cellulose, lignin, and hemicellulose. So I'll be talking a little bit of the digestive physiology of the cow. So right when it enters the mouth, we know it's going to be masticated, which means it's going to be chewed. And then that is to enlarge the surface area of the feed. So that is when the saliva would take place, whereby the amylase, salivary amylase, would turn the starch into maltose. It's actually okay. Pardon? Cellobiose or cellobiose in the mouth. Oh, okay. Okay. But we have two functions of saliva. Over here is called buffering, or it will suppress the foam to prevent from bloating. And then, as we know, it goes inside the rumen, and that's when the cellulose will be turned into the volatile fatty acids, which are acetic acid, propionic, and butyric acid. And then, what are the kind of anaerobes we have in the rumen are bacteria, fungus, and also protozoa. And then we have the pancreas secretion, in which it also contains the pancreatic amylase and the lipase, change the lipid into smaller forms, and then the intestinal enzyme for maltase, lactase, and sucrase. So that's what I focused on. So who wants to share next? Probably Shelly could, yes.
Okay, as you all know, cows depend on human microbes to convert feed components, in this case is carrot, into usable source of energy. Human microbes ferment carbohydrate to make volatile fatty acid and gases. Once the carrot is ingested, it is briefly chilled and mixed with saliva, and is swallowed and passed through the esophagus and into the rumen. The rumen is adapted for the digestion of fiber. Temperature inside the rumen is around 39 degrees Celsius, which is suitable for the growth of those microbes that I mentioned earlier. The microbes break down the feeds through fermentations. The pH of the content of the rumen and reticulum is maintained at 6 to 7. So, the chemical composition which found in the carrots, including soluble sugar, storage carbohydrate, structural carbohydrates. So I would like to talk about structural carbohydrates. The bacteria that digest structural carbohydrates such as cellulose and hemicellulose will produce a large portion of acetic acid, which is important in the production of milk fat. Whereas for the storage carbohydrate, the bacteria that digest the starch, which is a storage carbohydrate, are different from the cellulose digesting bacteria. They are insensitive to acidity and produce mainly propanoic acid. Last but not least, the soluble sugar, which is glucose, fructose, sucrose. The bacteria that ferment feeds high. These soluble sugars are similar to those that digest the starch. So the end product formed in the rumen, which is acetic acid, propanoic acid, which is known as volatile fatty acid, will then absorb through sources. Those energy is used to perform various functions such as milk production, maintenance of body system, pregnancy, as well as growth.
Just to add into some of the points mentioned by Aiman and Steve. Basically, in ruminants, they have a lot of bacteria in their rumen to digest cellulose. In general, the bacteria responsible for digestion of cellulose is Ruminococcus, and this bacteria produces cellulase, an enzyme to digest cellulose, and there is two type of enzymes involved. That is endoglycosidase, which cleaves the disaccharide cellobiose, which is an end product from the digestion of cellulose in the mouth. Then another type of enzyme is beta-glycosidase, which hydrolyzes cellobiose and cellodextrin producing glucose.
So, in response to what you found out, as you've said earlier regarding the composition of carrots, there's the free sugars and there's the cellulose and hemicellulose and the lignin. So when the cows consume carrots, for the digestion of cellulose, the process goes something like this. As you mentioned earlier, the Ruminococcus, it's a gram-positive bacteria, and it will degrade cellulose into the monosaccharide glucose in the rumen. And basically, it breaks the beta linkages in cellulose as it forms the glucose. And this mainly the primary carbohydrate site of, let me rephrase that. The primary digestion of carbohydrate is, it happens in the reticular lumen. So, after this degradation of cellulose, we get this glucose, which will then proceed to the glycolysis process. And I shall look into this.
What is glycolysis?
Okay. So basically, glucose is phosphorylated into glucose 6-phosphate, and this is aided with the enzyme hexokinase and the ion Mg2+. And next, these glucose 6-phosphate molecules are then converted into fructose 6-phosphate molecules with the aid of phosphohexoisomerase. Next, the fructose 6-phosphate is phosphorylated into fructose 1,6-bisphosphate molecules with the aid of the enzyme phosphofructokinase 1. Bear with me, this is very confusing. Next, fructose 1,6-bisphosphate is cleaved into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate with the aid of the enzyme fructose 1,6-bisphosphate aldolase. This dihydroxyacetone phosphate, it cannot proceed with the glycolysis in the subsequent glycolysis process, so it has to be converted into glyceraldehyde 3-phosphate. So this process occurs with the enzyme triose phosphate isomerase. So once they are converted into glyceraldehyde 3-phosphate molecules, they are then oxidized into 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Next, the enzyme phosphoglycerate kinase will then transfer a phosphate group from the 1,3-bisphosphoglycerate to ADP which will then form the product ATP, adenosine triphosphate, and the once 1,3-bisphosphoglycerate will become 3-phosphoglycerate due to the removal of the phosphate group. So, and next, the 3-phosphoglycerate will be converted into 2-phosphoglycerate, and this reaction will be catalyzed by the enzyme phosphoglycerate mutase with the aid of magnesium ions, Mg2+. Next will be the dehydration reaction of 2-phosphoglycerate to phosphoenol pyruvate. This reaction will occur with the aid of the enzyme enolase. And lastly, the phosphoryl group from phosphoenol pyruvate will be transferred to adenosine diphosphate, so it will form adenosine triphosphate. And the once phosphoenol pyruvate will become pyruvate. This all occurs with the aid of the enzyme pyruvate kinase, along with the ions, potassium ion and magnesium ion. So throughout this entire process, overall, one glucose will produce 2 pyruvate molecules, and 2 adenosine diphosphate molecules will combine with 2 inorganic phosphate molecules to produce 2 adenosine triphosphate molecules. So, how do we proceed from there?
Yeah, okay, my part is a continuation from your part, Ms. Nina. Okay, when cattle consume the carbohydrate, then the carbohydrate will go through many metabolic pathways, which is the one named DCA cycle, which stands for tricarboxylic acid cycle or Krebs cycle. It occurs in mitochondria. It is the central role in metabolism, and the certain reaction also occur in other metabolisms such as lipid and also the protein, because the fatty acid can be converted into acetyl-CoA, and also the amino acid can enter from many various pathways in this Krebs cycle. Okay, DCA is antibiotic, which means it performs either anabolism and also catabolism. Okay, the pyruvate from Jasmina part just now must enter the Krebs cycle, but then must be converted to acetyl-CoA by pyruvate dehydrogenase complex, which there are 5 of them. Next, the reaction occur, which actually has 8, which is the formation of citrate, isocitrate, alpha-ketoglutarate, succinate CoA, succinate, fumarate, almalate, and also regeneration of oxaloacetate, so that the cycle can be done for the next reaction. Okay, the end product for this Krebs cycle is the formation of 6 NADH, 2 FADH2, and also 2 ATP and 4 carbon dioxide, which is this pathway is actually aerobic respiration that yields carbon dioxide and also water.
Wow, so we're done. Okay, so what can we say about what happens to the cow? Anyone want to suggest any hypothesis here? The cow survived, or the cow obviously survived, right? And it lives with the carrot.
Yeah.
So that is all.